Good evening, everyone. We kept the best for the last. It's my great honor to introduce my Norotri brother and my teacher, Roozbeh Kiani. Roozbeh started his work in Dr. Esteki's lab. He's the first one to record from a primate in Iran, and he published quite well from his work in Iran. Then he joined Mark Shadman's lab to do his PhD, and he did tremendous work on the representation of confidence in the brain. And his thesis work was selected as the best thesis at the SFN that year. He then joined Bill Newsom's lab to continue his work with multi-array recordings in the primate frontal cortex, and he published very nice papers from that work. After his very short postdoc for a primate physiologist, he started his own lab at NYU, and he's been there, I think, more than 10 years, which he publishes, again, quite well. We have a saying in Mike's lab that any student is very good. We call him that he's the second coming of Roozbeh. And please join me to welcome Roozbeh, and I'm eager to listen and watch your talk, Roozbeh. Thank you for accepting our invitation.
Thank you so much for an overly generous and kind introduction. It's great to see you, Mehdi, and thank you everyone for attending the talk. As Mehdi mentioned, I'm coming from NYU, so it's customary here to ask questions during the talk. So if you would like to do that, please feel free. Otherwise, I guess I would wait until the end of the talk to get your questions. Okay, so my lab at NYU studies the decision-making process in humans and monkeys. And as many of you know, a hallmark of the decision-making process is its flexibility. So imagine me coming out of a poster session in a conference, like recently I was at COSI, and then I see a group of friends that are talking to each other, or a group of people that are talking to each other. And I need to decide, well, should I approach them to say hi and engage in the interactions with them or not? So I may decide that based on different factors that may be present in the scene that I'm seeing. One of them is the familiarity of the faces. Like, okay, is this group of people like a group that I'm familiar with and I want to interact with, or they are potentially new people that I don't know, but I would like to make friends with? Or I may make my decision based on identity. Like, is that person that I'm seeing here my colleague at NYU? Here are a few pictures for those of you who don't know who this person is, and you probably have recognized that this is like King Tony Mavshin. Or I may make my decision based on other factors. For example, what is the mood of the conversation that is going on? Is it like very intense thing that I should not approach, or is it like a lighthearted conversation that I can potentially join in? So I can make all of these decisions based on the same sensory input. So I see the stimulus, but depending on what my goal is and the factors that I may perceive, I may decide to approach the people or not to approach the people. Okay, so let me just jump to how we study these in the lab. Imagine a simple task that has been used extensively in the past for studying the decision-making process, where monkeys or humans observe a patch of randomly moving dots and decide, based on the perceived direction of dots, what action to generate. These type of experimental procedures has been used quite a bit for studying the decision-making process. I will just give you a short intro of them and the basic neural responses that are known about them, and then I will go into the more recent questions that we are asking. Okay, so in this case, the monkey observes the motion stimulus and then reports the perceived motion direction with a saccadic eye movement going to the right target for a rightward stimulus, or to the left target for a leftward stimulus. Because as the monkey performs this task, or as humans perform this task, we can also systematically change the strength of the stimulus from a strongly leftward stimuli to a strongly rightward stimuli. And we do that by changing the percentage of dots that move coherently together. If all the dots move towards left, that's going to be a very strong leftward stimulus. And if all the dots move towards right, that's going to be a very strong rightward stimulus. And in between, if different fractions of dots are moving in one direction versus another, that's going to be a different level of strength in the stimulus. So it has been shown many times by us and others that in tasks like this, as the strength of the stimulus systematically changes from left to right, the probability of making one of the choices, like the rightward choice, changes systematically. So the monkey is correct for the very strongest stimuli on both sides. That is, the probability of choosing the rightward target becomes 100% for a strong right and 0% for a strong left. And for intermediate stimuli, there is a less extreme distribution of the choices. Okay, so as monkeys perform this task, we can record from different parts of the brain. For example, in the direction discrimination task, we would like to study which parts of the brain represent motion. That's the sensory evidence that's used for the discrimination of dots in the random dots task. Okay, so I'm not sure if sound can be transmitted well through the link that we have, but I hope that you are going to hear it. Here is recordings from Hubel and Wiesel from many decades ago, where they are moving a light wand in the receptive field of cells in V1, and you can potentially hear the spiking activity of these neurons. Okay, so if you could hear the spiking activity, each spike is like a click. And then when the wand was moving in the receptive field of the cells towards bottom left, the frequency of those clicks increased, indicating that this cell likes a moving stimulus in that direction. But the tuning width of the cell is very narrow. A slight change in the direction of motion is going to substantially reduce the activity of the cell. Now, the motion selective cells in V1, which are a small portion of cells in V1, they project to regions in MT and MST in the temporal cortex of the monkey brain. And these are regions that are dominantly composed of cells with direction selectivity. Here are recordings from MST with the random dots stimulus in Bill Newsom's lab. And again, you are going to see that the random dots moves in different directions, and the frequency of clicks or the action potentials of the cell that they are recording changes based on that. Okay, so in the first part of the stimulus, the motion was towards bottom left, the frequency of clicks went up, and in the second part, the motion stimulus was towards top right, and the frequency of clicks went down a little bit. Okay, so these electrophysiological studies, in conjunction with inactivation studies and stimulation studies, have established that MT and MST neurons, as well as V1 cells, provide the sensory evidence that may be used in these type of decisions between right and left motion, for example. Okay, so here is better quantification of the responses in MT as the strength of the motion the stimulus is changing. So I told you that the motion coherence, the percentage of dots moving coherently, can be systematically changed. And when we do that, we see a systematic change, a quantitative change in the neural responses. When the motion stimulus is very strongly rightward, the rightward selective cells have very high responses, sustained responses throughout the stimulus, and the leftward selective cells have very low responses. For intermediate motion strength, the difference of the two pools of neurons is smaller, and for weaker rightward motion stimulus, there is still a difference, but this difference is even smaller. Okay, so now at each moment in time for a particular stimulus strength, the difference of the activity of the rightward and leftward selective pools of neurons can be modeled as a sample drawn from a distribution. So for example, for 6.4% coherence, this is the distribution that you see. The distribution, the bulk of the distribution is slightly towards right, meaning that the rightward pool is more active than the leftward pool. But at different times and different trials, you can also find situations in which the motion is rightward, but the rightward selective cells respond more weakly compared to the leftward selective cells, and the difference becomes negative. Now, the mean of this distribution is explained by K times C. K is a factor that we call sensitivity, and C is the coherence of the stimulus. This linear relationship is very important because it means that as the strength of the stimulus increases, the distribution shifts more and more towards right, because the difference of the activity of right and left increases. And this linear relationship could be used in a variety of model fitting approaches. And that's also one of the reasons that we like to use the random dots stimulus. And it has been so frequently utilized in the past because we understand so much the sensory properties of the neurons in MT and MST, and we can easily model them and model the behavior based on them. Okay. These neurons that are direction selective, they send their outputs to neurons in different regions of the brain that are involved in moving the eyes. In the task that I showed you, the response is generated by making saccadic eye movements. And for example, lateral intraparietal cortex in the posterior parietal cortex of monkeys is a region that is involved in planning the saccadic eye movements. LIP, as well as many other nodes in this network that is involved in planning and generating saccades, has interesting response properties. So first of all, these cells have visual selectivity, meaning that they respond or they increase their activity when a stimuli appear in certain regions of a space that is the response field of the cell. Second, these cells increase their activity when the monkey plans an eye movement to the same location in the space. And last but not the least, these cells have persistent activity. So when a stimulus is flashed on the screen and the stimulus is taken away, these neurons maintain the response and keep a memory of the stimulus. This can be nicely shown by a paradigm that is called memory-guided saccade task. In this paradigm, so if the shaded region represents the receptive field of the neurons that we are recording from, and the fixation point is where the monkey starts the trial, you're going to see that the target just flashes in the response field, then the target goes away, then after a delay, the fixation point is turned off, and at that point, the monkey needs to report the perceived location of the flash by making a saccade to that location. But of course, there is no stimulus there, so everything is done based on memory. Okay, so here is an example from an LIP cell recorded a while back. Okay, so the yellow thing in the middle was the eye of the monkey or the gaze of the monkey. You probably saw that a stimulus flashed very briefly there. Then the monkey maintained fixation and at the end made a saccade to the remembered location of the target. Now, specifically when the stimulus showed on the screen, the activity of the cell jumped to high and then it stayed high. So each click here is one action potential, which you could also hear in the audio. Okay, so it continued until the fixation point turns off and the monkey starts the saccade to the remembered location. Okay, now, if instead of flashing the stimulus in the response field of the cell, we flash the stimulus in a different location. Okay, so again, the monkey remembered and the monkey generated a saccade there, but the activity of this cell went below baseline and stayed below baseline. But presumably there are other cells whose receptive field overlaps with that other target, and those were the cells that increased their activity. So overall, the population activity within LIP at each moment in time is effectively keeping track of where is a potential saccade target and maintains the memory of it and ultimately helps with the execution of the saccade at the right time. Okay, so putting all of these things together, it makes sense to think about these cells as cells that gather information about potential targets of action, maintain the memory of them, and ultimately generate those actions or help with generating those actions. And specifically, they are distinct from sensory cells because in sensory regions, when you take away the stimulus, the activity of the cells dies down very quickly. Okay, so Mike Shatton a while back suggested that, well, this could be a mechanism that may help us to free ourselves from immediacy. So we have known for a long time that persistent activity may be important for working memory and maintaining information in memory. But in addition, like this can also be useful in cognitive tasks in which the subject or the agent has to operate on their own time scale instead of a time scale dictated by the environment. We can gather information, we can maintain that information in our mind, and we can act at the time that we deem suitable. Okay, so this freedom from immediacy is a key point in cognitive tasks, especially in decision making, and finding out what are the neural mechanisms associated with it are important, and the sustained activity could be one way that the brain can be contributing to it, or LIP can be contributing to it. Okay, so now that we know LIP cells have this interesting sustained working memory activity, maybe this property is also useful for decision making, because in the direction discrimination task, for example, the subject has to be integrating information over time. The decision about the motion and stimulus is not something that can be made in just like one frame of motion. Usually humans, monkeys look at the stimulus for some time, and after that, they know what the motion direction is. So this would require integration of information over time, which would exceed what I just showed you in the previous task. In the previous task, the location was maintained in memory, but that memory was not manipulated. For the direction discrimination task, information comes in, information is maintained, and then information has to be manipulated. That's how integration is done. You need to maintain the previous piece of evidence, then add the new piece of evidence, then keep the result, then add the next piece of evidence, and so forth, so on. Okay, so let's look at the activity of the LIP cells to see how they behave in this case. So again, this is the receptive field of the cell. And by the way, I call it response field from now on, because these are the cells that I think connect sensory stimuli to action, and therefore they are more than just receptors of the information. Also, you are going to see something interesting now, which is that the dots that are guiding the action, the saccade, are not shown in the response field of the cell, but rather they are shown outside of the response field of the cell. And yet, you are going to see modulation of the neural responses. So, okay, let's see what's going on here. Okay, so there were two targets appear on the screen, then the motion stimulus appeared. The motion stimulus appeared here, where my mouse is. The response field of the cell was here. And nonetheless, the appearance of the motion stimulus here and the movement of dots towards this location in a space increased the activity of this cell. After the motion stimulus went away, the activity stayed high, just like the memory-guided saccade task, and ultimately the saccade was generated. Okay, so when motion is in the opposite direction, what would happen? Okay, so this way, this time motion was in the opposite direction, the activity of the cell went below baseline, and the state like that, after the motion stimulus went away, they still stayed below baseline, and the animal generated the saccade. Okay, so basically what we are seeing here is that these cells that are representing this part of the space, or cells that have response fields in that location in the space, are not just sensitive to what happens in that location in the space, but they are sensitive to any information on the screen or outside of the screen that could potentially be used for guiding an eye movement to the location that these cells like. So this is one of the reasons that I call that location in the space that the cell likes the response field of the cell, as opposed to the receptive field, or maybe you want to call it the action field of the cell or something like that. So these cells connect sensory stimuli to actions, and they do that in a flexible manner. Okay, so let's, I showed you qualitative responses in two trials. Now let's quantify it a little bit better. If we record from a population of cells in region LIP as the animal discriminates moving a stimuli, we see the following pattern, that when motion is towards the RF and the monkey ultimately generates a saccade to that location, the activity of the cell would go up. When motion is in the opposite direction and the monkey generates a saccade in the opposite direction, the activity of the cell would go down. And these two traces that I'm showing are for very strong rightward and very strong leftward stimuli. We can systematically change the strength of the stimulus. And when we see that at about 200 milliseconds after a stimulus onset, there are traces that go up and down, but by different slopes. And the slope depends on the strength of the motion. When motion is towards the RF, the activity would go up, and for a stronger motion, the activity would go up faster. So this is also compatible with integration of information over time, because if you are sending more information, or if you're receiving more information from the screen, then probably if you're integrating, the integral should grow faster. Okay, these traces that are going up at different rates all come together at a common point immediately prior to the onset of the saccade in a reaction time task, for example. Then effectively we can think about that as a threshold that once achieved, a commitment is made and an action is generated. Because these type of neural responses in the DOTS task have been observed not just in the parietal cortex, but in a variety of cortical and subcortical regions. In frontal eye fields, which is another node involved in generating saccades, in a vast portion of the dorsolateral prefrontal cortex, in caudate or different regions of basal ganglia, in the superior colliculus. We have also shown that in motor cortex, for example, when the response is made by moving the arm, the activity very nicely recapitulates this integral, and so forth, so on. These results have also been replicated in rodents, although in rodents, the amount of cortex that is dedicated to this is substantially less than in monkeys. So these type of response properties, at least on face value, are connected to models, psychological models that have been proposed for how decision-making is done. In these models, the idea is that the stream of evidence, sensory information would come in. It's a noisy thing. It's noisy because, well, the stimulus itself is noisy, but also the representation of the sensory information in areas like V1, MT, and MST is noisy. So those regions provide moment-by-moment changes of the stimulus, and then those can be integrated over time, and the result of that integration process may be represented in LIP and other nodes that I just mentioned above, and the integration continues until the integral reaches a criterion that would trigger an action, just similar to what we saw in the LIP activity, that they grow and they reach a criterion, and once that criterion is reached, then that dictates, well, what is the choice? For example, if rightward selective cells win or reach their threshold, then it is the right choice. And the time to bound is effectively the decision time or the deliberation time that is used for making the decision. So this is more or less the current state of knowledge in terms of how these type of cognitive processes could be implemented. And a few a few mechanistic models and biophysical models have been proposed for how to implement this integration process. I don't go into the details of those models, but at the core, they share more or less the same idea. The idea is that there are two modules representing the competing actions, for example, the rightward saccade and the leftward saccade. And these modules compete with each other because they are laterally or maybe in a feed-forward manner. They are inhibiting each other. So the increased activity in one means that the other one has lower activity. And this competition between these two modules is biased based on the sensory information. So if sensory information comes in that more strongly supports a rightward action, then the activity of this module increases, the activity of the other module decreases, and then over time, a threshold is reached and a saccade is made. Okay, so if this is right, if this implementation is correct, then what really separates different decisions from each other is just what is being integrated, not the integrator. What do I mean by that? Well, in the direction discrimination task, it is the direction of the motion that we use in order to make a decision. But you can imagine an alternative task in which, say, the density of dots could be used, or the contrast of dots could be used, or you can just replace the motion stimulus with, say, a color stimulus, or a face stimulus, or an object recognition stimulus, you name it. No matter what the stimulus is and what the relevant source of information is, as long as it is the right saccade and left saccade that are competing with each other, then the integral or the decision variable, how much evidence is accumulated so far, should just be reflected as in the difference of the activity of these two modules, and that's it. Okay, so that's a very strong prediction, and this prediction has not really been tested up until we started approaching it. The reason that it was not tested very strongly is that recording from the monkey brain in just one of these discrimination tasks is challenging enough. So people usually just picked one task, recorded, and then made inferences and generalizations based on that, which led to the connection between DDM and the activity in LIP that I mentioned in the previous slide, and these type of mechanistic models that have been proposed for decision making. So Goki Okazawa, a brave postdoc in my lab, decided to really go after this prediction. And he trained monkeys to perform 3 different tasks: 1 direction discrimination, similar to what I showed you before, and 2 phase categorization tasks. So the direction discrimination task is very similar to the classic experiments that I talked about a couple minutes ago. Okay, so the monkey fixates. Here is the response field of LIP neurons. Then 2 targets appear. One of them is in the response field of the cells we are recording from, and another one on the opposite side of the screen. Then the motion stimulus appears, and just as past experiments, we can systematically change the strength of the motion stimulus on a trial-by-trial basis. And at the end of the motion stimulus, there is a delay period, and finally the fixation point turns off. That's the go cue for the monkey, and the monkey reports perceived motion direction by a saccadic eye movement. And if they are right, they would be rewarded. Okay, so this was the direction discrimination task. For the phase categorization task, all that we did was to replace the motion stimulus with phase stimuli. These phase stimuli are morphed. For example, they can be morphed between monkeys and humans. And the job of the monkey is on each trial to tell whether the stimulus that appears on the screen is more human-like or monkey-like, and would report the perception by making different saccadic eye movements. Okay, so just like the motion task, which has a stochastic stimuli, we also made our phase task a stochastic input task. And the way that we did it was by slightly changing informative regions of the face, the eyes, the nose, and the mouth from one frame to another frame. Each of these frames is about 100 milliseconds long. And at the beginning of the frame, a face stimulus appears, followed by a mask. And then when the next frame is shown, some of these features are modified slightly. The masks would prevent the subject from knowing exactly which feature has changed. And the changes are small enough that the perception of the subject is, well, there's a face there and clouds are coming and going. I'm going to show you a movie of that in the next slide. But we, the experimenters, can show that those tiny changes in the features of the face are actually informative and can tell us how the subject weighs information in different regions of the face in order to make a decision. Okay, so in addition to the identity task, the monkey-human discrimination, or occasionally human-human discrimination, we also had an expression task for the monkeys. In the expression task, it was just the expression of the face that would change from sad to happy. I'm using labels sad and happy not to imply that the way that monkeys perceive expression and emotion is similar to what humans do. But from a variety of studies, we know that they are very good at noticing changes in facial features. And sad and happy are just names for us, labels for us to be able to talk about the stimulus. Otherwise, I'm not implying anything more than that. Okay, so the stimuli are effectively more or less coming from the same space. So you probably have noticed that the sad, happy are really changes that are happening on the 0% morph on the identity line. So you can imagine a 2-dimensional stimulus space that can be divided based on one category rule or another category rule, and then the subject would be instructed to discriminate either the expression of the face or the identity of the face. So here is the movie that I promised that I would show you. So this stimulus here is just a morph between my face and Goki's face, and probably your perception, I mean, unless you're super good in noticing the small changes, your perception should be that, well, this is more or less like the same face, but some weird type of mask is coming and going like a cloud on it.
Sorry, sorry, we have a technical problem here.
Sure.
We lost your screen and your camera feed. If people here can fix it soon, that would be great.
Okay. Let me. Should I try to reshare my screen, or should I just wait for you?
I think the problem is in our side, not yours.
Okay.
I think we can see your camera feed. I'm not sure. Is it the scene? Is your
Yeah, I stopped my slide, but I can.
Yes, I think you're back. Thank you.
Okay. Do you see the movie playing on the screen?
Yes, yes, we do.
Okay. So, sorry everyone for the technical challenge, but I guess it is to be expected given the internet speeds and other problems that may exist. Okay, so as I mentioned, and let me just quickly recap, from one frame to another frame, we are changing the features of the face by just tiny amount. And because there are masks, you probably are not noticing that those features are changing. So those changes are subliminal because you're not perceiving them. But nonetheless, if you do the task, these tiny changes are in fact influencing your choices. And this is what we use in order to know how you would weight the information. Okay, so let me just go into the engineering of the stimulus, because it may be important for understanding what to come. The face stimulus is, of course, a very complex stimulus with a lot of things in it. In order to control the complexity, we reduce the dimensionality of the stimulus by focusing on regions that we define as informative. So specifically for each prototype face, and by the way, the monkey performed many of these different identification tasks or expression tasks. So for each prototype face, we choose an eye region, a nose region, and a mouth region. Everything outside of these regions are always fixed at 50% morph between the two prototypes, but the three informative regions can be independently modified. So now we have reduced the dimensionality of the face from a very large, like pixel space into a three-dimensional feature space. And what we can do now that we have engineered the stimulus this way is that we can independently morph different features. Like, for example, I can create a stimulus that has eyes more similar to monkeys and mouth more similar to humans and nose that is in between. And then in the next frame, these things can change again and again and again. And by these changes and checking the impact that they have on your choices, I will know how you are weighting different locations in the face in order to discriminate faces. And here is a two-dimensional stimulus that I alluded to. In this two-dimensional space, the identity is changing across one axis and the expression is changing across another axis. And you can imagine redefining category boundaries for identification and for expression discrimination. The monkey can be trained to do both of these tasks, and then on different sessions or on different blocks of within a session, we can ask the monkey to perform one or another. This is important because now we can fix the stimulus to the same thing, but apply two different sets of rules on it. Okay, so I told you about the design of the task, but can monkeys do these tasks? The answer is yes. For the motion task, of course, we knew that they would be able to do it. Here is the probability of generating a correct response as a function of the stimulus strength. And you can show that for a strongest, we can see that for a strongest stimuli, the monkey is almost perfect. For weakest stimuli, the monkey goes down all the way to chance. This is the psychometric function for the face task, and again, the same transition from chance to perfect accuracy is observed. One other thing that we can do is we can systematically change the duration of the stimulus from one trial to the next, and we can measure whether those changes facilitate the behavior of the monkey or improve the accuracy of the monkey. The idea is that if the monkey is using information across the stimulus presentation, then the monkey should perform better for longer stimuli compared to shorter stimuli. We can show that by looking at the changes in psychophysical threshold as a function of a stimulus duration. Psychophysical threshold is defined as the stimulus that generates 81% accuracy. And the idea is that if the stimulus is longer, then a weaker stimulus may be sufficient through integration of evidence to generate the same accuracy. So for longer stimuli, we indeed see that the psychophysical threshold or the stimulus required for that accuracy level would go down, indicating that subjects are really not making their decision based on one snapshot of the stimulus, but rather they are integrating information across the stimulus. Okay, so that's about motion, and the same thing is observed in the face task. Okay, so that was the comparison of motion and face tasks, but I also told you that we have two different types of face tasks. And the way that we can, let me just jump to that. Okay, I told you that we have two different face tasks, and we can look at the performance of the monkey in those tasks. So here is the identification task, so psychometric function, expression task, psychometric function, and the thresholds associated with them. So threshold going down, indicating that some form of integration is taking place. So very little work has been done about how these sensory decisions are made with complex stimuli, for example, like faces. And here I just want to show you that a simple extension of the accumulation to bound model that I showed you earlier should be adequate to capture the behavior of the subjects here too. All that it takes is to imagine that, okay, the mouth region, the nose region, and the eye region are all providing information. At each moment in time, we can combine them linearly or non-linearly. Here, I'm just focusing on the linear integration across a space. We can linearly integrate them. Now we have momentary evidence from the combined informative features. And then those momentary pieces of evidence can be integrated over time to create the decision variable. And again, when the decision variable reaches a threshold, that terminates the decision-making process and a choice is generated. Okay, so when we do that, we can go back to, for example, the face task, or the two face tasks, identification and expression, and ask how the weights change from one task to another task. For the identification task, it is the eye region that is most informative. For the expression task, it is the mouth region that is more informative. And these changes in the informativeness of the features for the two tasks also match previous psychophysical studies in humans in different paradigms that define, okay, like eyes are informative for identification and mouth for expression discrimination. Okay. Because I don't have like a lot of time to go into the extreme details of these modeling piece, I would just list quickly a few properties to, again, establish match between the face task and the motion task computationally. Okay, so we can calculate what is the amount of loss of information in the temporal integrator, a term that is called leak. And we can show that the time constant of the integration process in both tasks is quite large, about 20 seconds or higher, and that means there is minimal amount of loss of information in either task. So this is encouraging. The other thing that we can check is, well, in the motion task, people have shown that the weights on the sensory information remain constant over time. For the face task, we can do the same thing. We can check whether the impact of different features would change as a function of time. And the answer is that not really. We don't see any meaningful change in terms of time dependency of the spatial integration process. And finally, we can check whether the spatial integration should happen prior to the temporal integration or it can happen after temporal integration. We can, for example, imagine a system in which separate decisions are made based on eyes, nose, and mouth, and ultimately there is a voting process that would lead to the perception. But we can rule out that final process, and it is really integration over a space followed by integration over time, which is almost identical to what people have shown for the motion task. Okay, so from a computational perspective, and based on behavior, as much as we can tell, these tasks are more or less similar to each other. But how about responses of neurons in LIP? Here I'm showing you the average activity of the LIP cells in the motion task. And similar to what had been shown in the past, we see a systematic stratification of the activity of the neurons based on the motion stimulus. So when motion is strongly supporting the saccade to the RF of the cell, the activity of the cell is higher, and when motion is strongly in the opposite direction, the activity of the cell is lowest. For the phase task, we see a reversal for the firing rate associated with making a saccade to the response field of the neuron. So this was surprising because effectively what I'm showing you now is that the strongest phase stimulus that supports a saccade to the RF of the cell generates actually the weakest activity among the rightward selective stimuli. And the strongest activity is actually generated by a stimuli that supports that action most weakly. This reversal was a little bit puzzling for us, so we spent a little bit of time to make sure that this is a robust finding. We recorded from different regions along the interparietal sulcus. We focused on cells that have memory of selectivity or they don't have memory selectivity. We checked whether any overlap between the stimulus and the RF of the cell may be responsible for this reversal. But the answer for all of those things is that, well, not really. This is robust and it can be replicated in a variety of locations along the interparietal sulcus. It can be replicated for different subpopulations of cells in a lateral interparietal region, and it's also not due to overlap between the stimulus and the RF of the cells that we are recording from. Then we said, okay, now that this finding is robust, instead of just averaging the neural responses as people had done before, how about we look at the population activity? There are a large number of ways that it can be done. The simplest way is probably looking directly at the status space. So the status space can be defined by the firing rate of different neurons. For example, if you are recording from n neurons, you can imagine an n-dimensional space. And the activity of the population, the n neurons at each moment in time, is really a point in this space. So we have different stimulus strength, so we can pick a point in time, and we can ask, well, for different stimulus strength, what the activity of the population is. And in order to visualize it for you, I will just use simple dimensionality reduction and the principal component analysis in this case. So when we do that, and by the way, these like three-dimensional PCS spaces that I'm showing you are explaining more than 50% of the variance. So the patterns that we are seeing here should be taken reasonably seriously. Okay, so for the phase task, as the activity goes from supporting a strongly leftward saccades to a strongly supporting rightward saccades, you see that the state of the population systematically changes, not on a straight line, as you may expect from linear attractor dynamics, but on a curve. And for the motion task, the same transition takes place. So, and by the way, these principal components are all coming from the mean responses of the cells to different conditions across the full stimulus viewing duration. Okay, so these curved manifolds are encouraging, suggesting that the population of the LIP cells are really separating different stimulus strengths and different motion directions from each other. They also suggest that integration over time is implemented in the population. The simplest way perhaps to do that is to look at the population activity as a function of time, and this is what I'm showing you now. At about 200 milliseconds after a stimulus onset, the population activity is really not separable for different stimulus strengths. But as time passes by, you see that the curved manifold starts to emerge and it expands. Okay, so this effectively suggests this is the signature that you would expect for the integration, and it effectively suggests that in both tasks, more or less the same thing is taking place. So the population perspective is compatible with integration of information and gradually improving separation of different patterns of the stimulus. But what happened to the reversal that we saw when we averaged the neural activity as people used to do for LIP in the past? So the averaging is really a vector in the status space, a 1-1-1 vector, so equal weight to all neurons. And because PCA is a linear dimensionality reduction, then a line in the n-dimensional space also would remain a line in the principal component of the space. And then I can look at the relationship of the curved manifold that we discovered with respect to this averaging vector. Okay, so the points that are along the manifold, when we project them on the vector, and these are orthogonal projections, but because we are transitioning from a 3D space to a 2D projection, they are not orthogonal angles, but believe me, they are orthogonal. The math is right. Okay, so when we do the projection, we see ordering that is of this form. So for the stimuli supporting the leftward saccade, you see a gradual increase of the firing rates, the way that you did for the motion task. But for the stimuli that are supporting the rightward saccade, there is a reversal. So the strongest projection on the averaging vector is created by the weakest stimulus. So that would lead to the reversal that we saw for the averages. And when we do the same thing for the motion, now we realize that the orientation of the curved manifold with respect to the averaging vector is different. So now if you rotate the curve and then project, you can end up with monotonic ordering similar to what we saw in the motion task. Okay, so this is interesting. Really, the bottom line about how the reversal takes place is that the curved manifolds that are representing the integration process are not fixed in the status space, but rather they rotate from one task to another task. And then we wanted to know why this rotation takes place. Because one way to think about it is that, well, in the phase task, you're using the phase stimuli. In the motion task, you're using the motion stimuli. Motion and phase are represented in different parts of the sensory pathways, motion in the dorsal visual stream and phases in the ventral visual stream, and maybe projection from different regions into LIP have different manifolds or are associated with different manifolds. I don't think that is totally true, because I told you that we used two different versions of the phase task. And now we can record from the same neurons as the monkey transitions from one task to another task. And we can have the same principal component axis for the two tasks. And then we can ask, well, what happens to the manifolds? If it is really differences in projections from different sensory regions, then you would expect the curved manifolds to be the same for the two phase tasks. But that is not true. The curved manifold actually rotates in a status space for the two phase tasks too. So it seems to me that the rotation is at least partially explained not by sources of information, but rather by the rule, or the rule that the monkey uses for making the decision, or the strategy that the monkey applies to a decision-making process. So now let me loop back to the key point that I started the talk with, that what does it mean? So we started by saying that the prediction of the standard biophysical models or mechanistic models is that the integrator should be more or less the same. I showed you that that may not be the case, and there is a rotation. And this rotation has implications. So let me highlight two potential implications. So one implication is that the integration process and also the readout associated with the integration process are task dependent. So I told you that in the phase task, we have like two different manifolds that are slightly rotated and modified with respect to each other. And we can ask a very simple question. We can say, well, if I find the best axes that represent the decision variable in the status space, or the integration of evidence in the status space for the two tasks, and then I use the readout axis for one to decode the decision variable for the other, what would happen? Because one possibility is that, well, you may have rotation in the status space, but this is a rotation that is not impactful. That does not seem to be the case, because when we do the manipulation that I just mentioned, or the game that I just mentioned, for example, I use the readout axis for the decision variable in the expression task, and I try to predict the decision variable for the identification task or the species task. There is a systematic bias, meaning that if I was using just one axis for readout, then I would have predicted biased choices in the species task compared to the expression task. But of course, the behavior of the monkey is not biased, meaning that whoever that is listening to the activity of LIP and is reading it out in a task-aware manner. Because the second point that I want to highlight, which is perhaps even more important than the first one, but also goes against the common intuition about how LIP and decision-making process works, is that the curved manifold actually indicates some form of dissociation between integration of evidence and action encoding. So here's again the curved manifold and the ordering of the average PSDHs. These patterns that separate from each other in the status space and the average activity during the stimulus viewing would actually converge to specific locations immediately prior to the saccade. So the data points that are associated with saccade to the T-in are collapsed to this location in the space, or the T-out collapsed to this location in the space, and the PSDHs also fall on top of each other. Okay, so the key point of whether integration process is implemented through difference of the activity of rightward and leftward selective cells is whether this curved manifold is aligned with this line in the status space. And that does not seem to be the case. There is misalignment, and that misalignment exists both in the high dimensional and in the low dimensional projections that we may make. Here I'm showing you the three-dimensional projections, and even in a three-dimensional projection, there is an angle. This angle is slightly smaller on the motion task compared to the phase task, but it is present in both. Okay, so now let me zoom back to the models. We started by saying that, well, maybe right and leftward pools of neurons compete with each other, and the difference of the activity implements the decision variable or represents the decision variable. Now I'm showing you that at least in the earliest stages of the decision-making process during a stimulus viewing, that is not the case. Pools of neurons can be competing with each other, but those are the pools of neurons that compete with each other are not necessarily the rightward and leftward selective cells. As the decision-making process continues and the time for action comes, now there is a rotation in the status space to align the representation of the integration of evidence with the representation of the action. And we see the separation of the activity of rightward and leftward selective cells only when we get closer to the time of the saccade. Okay, so if that is true, then the variety of models that have been proposed for the implementation of the integration process and decision-making probably need to be revised. Okay, I want to use maybe the next 3 minutes to just walk you through an interesting observation that we had here that may be of interest to you. And it is about the curved manifold. So the curved manifold, as you may remember, has effectively two key axes. One axis that represents the representation of the decision variable, so that it's along the manifold like this from one end to the other end, and you can imagine a linear projection or a linear axis that best represents that transformation. But there is also another dimension, this dimension, that is systematically changing with the stimulus difficulty. So at the peak or at the apex of the curve, we see the weakest stimuli, and at the base of the curve, we see the strongest stimuli, both rightward and left. And maybe this stimulus difficulty axis is something that is functionally important. So that functional importance comes down to another topic that I'm very interested in the decision-making process about the representation of confidence. So previously, in a previous paper that Mike Shadlin and I published, we had argued that it is the representation of the decision variable that is informative for the confidence that the subject has about the choice. But if there is an independent axis in the status space that is representing a stimulus difficulty, then maybe representation of confidence can just be dependent on that instead of dependent on the decision variable. Okay, so we decided to test this and go back to kind of the older paradigm that we had, where we trained monkeys for a post-decision wagering task and looked at the activity of the LIP during post-decision wagering. So here is the basic of the post-decision wagering. The monkey observes, again, the two targets. One of the targets is in the RF. Motion stimulus comes in, then there is a delay period. Then for some trials, in the middle of a delay period, we show a third target to the monkey. This target can be called a short target or a short bet option. And by choosing it, the monkey can always receive a small but guaranteed amount of reward. Okay, so at the end of the delay period, the fixation point turns off, and now the monkey could choose any of the three targets. If the monkey chooses the target that is correct, for example, the one in the RF, the monkey would receive a large amount of reward. If the monkey chooses the motion target that is wrong, the monkey would receive no reward. But the monkey can also opt out of the decision-making process and choose the short bet option for a small but guaranteed amount of reward. So when should a decision maker exercise this short bet option? And ideally, a decision maker should do that when they are not very confident about their choices. If the monkey is very confident about the choice, they go to the motion target that they think is correct. But when their confidence about the choice would come down, would decrease, for example, for weaker stimuli, then the monkey should start exercising this short bet option more often and settle for a small but guaranteed reward. And in fact, this is what we see experimentally, that the probability of opting out decreases systematically with the strength of the stimuli. For a stronger stimuli, when the monkey is confident, the monkey does not use the opt-out very often. And for the weakest stimuli, the monkey uses it more often. Okay, so first of all, the stimulus difficulty axes and the decision variable axes that I showed you about are useful for decoding the strength of the stimulus and correct and error and all of those things from the activity of the LIP cells. Here is the difficulty. So the stimulus difficulty axis could in fact be used for separating difficult stimuli from easiest stimuli in both tasks. But this axis does not seem to be very informative about whether the monkey chooses the short bet option or does not choose the short bet option. For the short target, we see that the activity is really not very much discriminable from each other. So that means that where you are on that axis, when you conditionalize on a particular stimulus strength, is really not predictive of whether the monkey is going to choose the short bet option or not. On the other hand, when you look at the projections along the DV axis, along the decision variable axis, you see that they are informative and they are predictive. Okay, so I kind of, this was a relatively, it was a distinct segue into the talk, so not related to the main topic of the talk. But I'm highlighting it just to mention one thing, that when you are working with high dimensional spaces and you find axes that are informative, you should be very careful about interpreting them. Because an axis like the stimulus difficulty axis in the curved manifold that I mentioned does not seem to be functionally important, at least in the confidence post-decision wagering task that we have. It may be used for other things, but not for here. So we need to resist the temptation of finding some decodable axis in the state space and then putting functional labels on it. So I think like that's something good to keep in mind as these techniques are used more and more often in the field. Okay, so the curved manifold that I showed you in LIP does not seem to be limited to just LIP. We also recorded in the motion task from two other parts of the brain. One of them is the prearcuate gyrus and the lateral frontal cortex. We also recorded from the supplementary eye field, which is on the dorsal part of the medial frontal cortex. And in both of them, we see the curved manifold. So again, like the prediction of the earlier models was that line attractor dynamics or integration can really be represented as a line in a state space, but we see ubiquitously across the cortex or across at least the frontoparietal cortex that it is really not a line, but rather a curve in the state space. Because let me summarize and highlight some of the important things that I told you. In terms of computational properties, face discrimination, and I also believe other object discriminations are compatible with the integration of evidence over time, except that there is probably some spatial integration happening prior to the temporal integration. And neurally, oculomotor planning regions encode the decision variable in both face and motion tasks, but they do that along curved manifolds as opposed to straight manifolds. And these curved manifolds are rotated with respect to each other, suggesting that the implementation of the integration process is not fixed, but rather task dependent. I showed you that the readout is task dependent, and I also showed you that it's really not the competition of action selective modules that implements the decision variable. We need to expand that perspective and think about more abstract type of representations. So I want to end by speculating maybe for a minute or two about how the curvature comes to exist. So I told you that the curve representation is ubiquitous, but what does that exactly mean? Why we are seeing it so frequently? I really don't know the answer, but I think the following lines of thought make sense to me, and I would love to hear your thoughts on this. So again, imagine that you want to implement some integration process through line attractor dynamics in an n-dimensional status space. The most straightforward thing that you may want to do is just to create a line in the status space that would represent the integration or gradual change of the decision variable as a function of the stimulus strength. In reality, however, you need to take into account some constraints. For example, the firing rate constraints. So we know that firing rates of the cells can't go below 0. We also know that high firing rates are energetically expensive and therefore probably are going to be avoided. And putting all of these things together, the part of the status space that you can actually use is limited. Now you have to fit your line in that limited status space. You can do that, but when you do that, the points that are representing different levels of decision variable and different stimulus strength would come close to each other. Another disadvantage that you have is that this line would effectively go through the center of the hypercube, where the majority of the neurons are showing more or less large amounts of activity, and this is something that you want to avoid. So you don't want the average activity to be very high for the weakest stimuli where you're least likely to be rewarded for. Okay, so if you want to take these two constraints into account, then maybe one way of doing it is to just get the center of that line and drag it to a corner, ideally a corner that is closer to the origin. And now what happens is that you have avoided the center part of the cube, which is energetically costly, and you have also separated your patterns from each other, the patterns that are representing different decision variables from each other, improving your decoding. Okay, so well, if curvy things are good, why limiting ourselves to a simple curve? Why not creating like a very twisted curve? A twisted curve would give you better separation of the neighboring points if you really can trace a twisted curve in the high dimensional space, but probably there are limitations, because when you do that, then points that shouldn't really be close to each other come very close to each other in the status space, and that would create challenges for the readout. So probably you want to have some amount of curvature, but not like very twisted, tangled curvatures. Okay, so if that is true, then maybe we can implement these insights in a recurrent neural network. So previous studies had shown that recurrent neural networks would give you actually line attractor dynamics as a line represented in the status space. But we said, well, let's get those, but now add a little bit of cost in terms of the number of spikes that the network can generate. So with in a collaboration with Christian Mawkins and Alan Manku, a former graduate student in Christian's lab, we implemented the spiking neural networks, and we added cost of the spiking to those. And immediately what we got out of it was a curved representation similar to what we saw in LIP, supplementary eye field, and the lateral frontal cortex. Okay, so this is how we think at this moment about the potential reason that curved representations exist. However, why rotations exist, why the same curved integrator cannot be maintained across different tasks, and we observe a rotation in the status space is a mystery. I have ideas of why we observe this mystery, and I'm happy to discuss them if you're interested, but I stop here to make sure that I'm not over time. Let me finish by thanking the people in my lab, especially Goki Okazawa and Christina Hatch, who were deeply involved in collecting the data, analyzing and modeling, as well as our collaborators, Christian Markens and Alan Manku, and the funding sources that made this possible. So thank you all. I stop here and take questions.
Thank you very much, Roozbeh, for this clear and great talk. If anybody has questions, which I really encourage you to ask your questions from Roozbeh, please raise your hand. And okay, the first one, Suda goes first.
Sorry, I'm not hearing you.
This take.
Oh,
Okay.
sorry. Hi, dear Dr. Kiani. I'm so pleased to hear your voice and your talk. Actually, the first time that we encountered your paper, it was very interesting and it was full of surprises for us on how you can approach decision making and very neuroscientific problem from a mathematical and geometrical perspective. And when we put these geometrical perspective, it all makes sense. You know, when we talk about the manifold and how it's rotated, it all makes sense. But when I think about the network behind it, when I think about how we can interpret it in the way that the rotation happens, what happens in the brain? What does what is the transformation happening in the maybe nodes of the circuits which make this thing possible? Maybe I'm asking this due to my lack of knowledge in mathematics and neuroscience, but I would be glad to hear your insight on this matter. Thank you very much.
Thank you. That was a great question. We have the same questions too. So that was the last thing that I mentioned in terms of how these rotations can be implemented. So there are two ways to approach this. We are in the thick of it. I don't have a clear-cut final answer for you, but I can share where we are at this moment and where our thinking is. Because there are two ways to approach this. One of them is that this rotation is actually a feature. That is, the brain is trying to create rotation in a status space, and that rotation in a status space is something that the brain takes advantage of. Another way to think about it is that, well, this rotation may actually be a bug. Maybe a bug is not the right adjective, but I think it captures the essence of what I'm going to say. So think about it from the perspective of the final nodes in the brain that are implementing the eye movements. Like something should send action potentials to eye muscles and then twitch those eye muscles so that my eye will move from one gaze to another gaze. For those cells, probably it is unimportant what the source of the eye movement is or why the brain is implementing the eye movement. So ultimately, at the end, what I'm trying to say is that at the end of the hierarchy of brain regions that are involved in the decision-making process, the information about task and context and all of those things probably eliminated. I don't expect that if I go to the abducens nucleus of the brain and I record from them, I would be able to discriminate the motion task from the face task. How the brain can actually generate a code like that is a challenge, because It's a recurrent network. It's a recurrent network with multiple circuits. So I told you about LIP, FEF, lateral and medial frontal cortex, basal ganglia, superior colliculus. All of these regions are connected to each other, usually reciprocally connected to each other. Within each of these regions, you also have recurrence. And that would mean that when information comes in, it's very difficult to get rid of it or to create action signals that are context independent. Now, let me create also an analogy to what happens at the level of ventral visual pathway. So keep what I just told you in mind, please don't forget it. And now think about like the deep nets. The deep nets, the stimulus comes in at a pixel level or say in the brain, patterns of activity of the retinal ganglion cells. Those patterns are all entangled with each other. And then the brain would require multiple stages of processing. And those multiple stages of processing would ultimately lead to separation patterns of activity associated with different categories and object identities from each other. Could the brain have done that in just like one shot from retina to LGN? Probably not. Like the brain is taking multiple steps in order to shed information that is not relevant and create a linearly separable representation for things that are relevant. For example, category membership or identity of the stimuli. Now let me come back to the part that I asked you to keep in mind, that these tangled representations that we are observing in LIP and the action-only representations that are probably represented multiple steps downstream to LIP can be thought of as something very similar to a brain attempting to separate different pieces of information from each other, shedding irrelevant information and keeping relevant information. So maybe LIP is somewhere in the middle of the stream of the processes that are necessary to connect a stimulus to action. And that is why in LIP, you don't have the action code fully separated from context and rule code. But as we move forward across the hierarchy of regions involved in generating the action, or across time within each of these regions, we see that the context that is causing the action is shed more and more, and the action code dominates. Like even within the LIP, I told you that at the beginning, during the stimulus viewing, you see the representation of context and the decision variable. But at the end of the trial, even within LIP, all that you really see is the representation of the action that would be generated by the monkey, right? So think about it like a deep net, or at least this is how I'm thinking about it. I may be wrong. I'm telling you that I'm speculating. It's full of speculation, and we are in the middle of like figuring it out. But the way that I think about it is that it may be similar to a deep net, but a process that is represented in a network of recurrently connected circuits that is unfolding over time. And there is transformation of the code within each of these regions and also across these regions as we get closer and closer to the final stages of the action generation. Does that make sense? So it's like if you think about it this way, then information would come into LIP, and then LIP may want to have one integrator that represents everything, but it just can't because of the constraints associated with the recurrence. And that is something that the circuit tries to figure out over time and over different nodes engaging processing.
Thank you very much.
This is the bug perspective of this of the answer.
I have to go and rethink and reflect on it. But
Sure.
For now, thank you very much.
You're most welcome.
Nick, does anyone have? Okay, Amir Reza has a question. Go ahead.
Okay, hi. Thanks for your great talk. I had two questions. The first one is that, so, we can say that you are capturing something in the phase task that, or we cannot, we cannot capture it with simple averaging and use population coding. Do you think if there is anything in the RDM, in the simple RDM task, that we cannot capture with simple averaging in these last 20 years? and we should look at the population level activity to capture that. So this is my first question, and the second question is actually some kind of hypothesis. So I think if we simply stimulate the LIP in the phase task, I think we should not see the simple bias in the behavior, because actually, the activity is reversed for the choice, with the presumption that stimulating is just, hiding the activity. Thanks.
Okay, thank you. Both of them were good questions. Let me start with the first one. So the first question was whether, okay, you guys froze for a second, but now I see you again. So the first question was whether averaging in the direction discrimination task is a good thing to do. Look, as you mentioned, it has been done for a long time. And if all that you want to get from the population is some readout of the decision variable, it's an okay-ish thing to do. There are better versions of the decision variable that you can still extract from the population. So as I showed you, even in the dots task, let me try to share my screen one more time to see if it works. Okay. Give a second. Okay, so even in the dots task, the difference of the activity of rightward and leftward selective neurons is really not aligned with the representation of the decision variable. Now, of course, if you, as I showed you earlier, if you project this curve on the line, you are going to get monotonic ordering of the data points, and you can get some readout of the decision variable, but this is not really the best readout of the decision variable. A second related thing is that this subtraction of the rightward and leftward selective or averaging perspective, which is the twin version of it, is has masked a nuisance or a nuance that exists in the decision-making process, which is that thinking about pools of neurons that are right and leftward selective as main things that compete with each other for the implementation of the decision may not be the best possible thing. It may be still productive to think about it that way, but there are strong constraints, and the fact that you cannot really generalize this perspective from one task to another, I think, is a major challenge. So if you are an engineer and all that you want to get is some representation of the decision variable, you can do that. But even as an engineer, you can do better. And if you're a neuroscientist and you want to learn about the neural mechanisms, then the averaging can potentially mask important details. Okay, so that was the first part. The second part, or the second question that you asked, was that the common readout does not lead to bias in the phase task. And yes, that is true. The common readout does not lead to bias in the phase task, but it damages you on a much more important front, and that's the sensitivity. So if you just look at the difference of the activity of right and leftward selective cells in the phase task, you are going to be punished very severely because the difference of the activity would be independent, more or less, of the strength of the stimulus, meaning that you would predict a flat psychometric function more or less at chance, regardless of the stimulus strength. But of course, this is something that we don't observe in the monkey's behavior. The monkey's behavior systematically improves for a stronger stimuli. Let me see if I can show you that slide. So again, I'm going to try to share my screen. And yeah, here it is. Okay, so let me imagine that for the motion task and for the phase task, I just look at the difference of the activity of right and leftward pools of neurons for a strongest stimuli to the weakest stimuli. Okay, so the strongest stimuli in the motion task, for example, would be 50% rightward minus 50% leftward. And you see a rise. And you also see nice stratification. So as you expect, for the weakest stimuli, patterns of activity are least separated from each other, or the averages are closer to each other. And for the strongest stimuli, they are best separated from each other. When you do the same thing in the phase task, you really don't see anything. You don't see separation by the stimulus strength, meaning that your decoding ability doesn't really improve with the stimulus strength if you use the averages. And just to demonstrate that, so here is the area under ROC, the receiver operating characteristic. And for the motion task, the area under ROC for individual neurons systematically improves with the stimulus strength. For the phase task, very little improves. So if you really think about the decision-making process as difference of right and left, and you want to predict what the psychometric function of the monkey would be, here you would predict a nice psychometric function. Here you would predict a very flat psychometric function, but that is not true in reality. Does that answer the question, or there is something that I missed? Okay, great.
Thank you. I think, okay. Setara,
Can I ask? Oh, okay. So, hello. Thank you for your presentation and also answering all of the questions. I have a question about what Sude earlier asked about that we don't know what is exactly happening in the brain. So, is it possible for our future works to somehow deactivate those regions that you told us that they are entangled, and then by recording one by one of them, we can not simply, but we can understand what is behind that? Can we?
Yes, and I think that's a great idea. This is a research program that we are pursuing, as well as many others. So most of the experiments that are happening in the lab at this moment involve recording from multiple of these nodes simultaneously, and then tampering with one of them and seeing how the activity in the other ones change. I think this is one of the most powerful ways to get at the bottom of this problem. Whether we are going to succeed or not, that is, whether these experiments are going to give us the insights that we need to figure out what is really going on in the brain, as you said, remains an open topic. For now, this is among the best ideas that exist in the field, and we and many others are pursuing, and I encourage you to do the same thing if you can. But I can't promise you're going to succeed. So.
Thank you.
Reza has a question.
Hi, Roozbeh. Hi, Roozbeh. How are you? Very interesting talk. I'm going to ask a question, but maybe I have misunderstood the whole thing. So correct me if I'm wrong. So for visualizing the shape of the manifold, you have used PCA, yes? Could it be that if you use another dimension reduction method, you will get completely different shape for the manifold and it's not curvy and so on?
Hi, Reza. That's a great question. So I showed you the principal component results because they are the simplest, and that's a technique that most people are familiar with. But we have played with a variety of dimensionality reduction techniques, linear and nonlinear. You can also try to characterize the shape of the manifold in the full dimensional space. You really don't need to do dimensionality reduction. So you can just ask, well, how the distances between neighboring points in the high dimensional space is changing. And then you say, okay, now if instead of looking at the neighboring, I look at 0.1 and 3.2 and 4 and so forth, so on, and then increase your gap systematically. So once you have all of these distance matrices, you would effectively get a qualitative picture of the shape of the manifold. And you can also get a quantitative picture of the shape of the manifold in the higher dimensional status space. And all of them point at the same thing. It's a curved manifold. The only thing that would change as you use different techniques is the dimensionality of this curved manifold. So the curved manifold that I showed you I showed you in a 3-dimensional space. We are analyzing it like more or less in a 10-dimensional space most of the time. And in that 10 dimension, we can show that it's probably like 1 to 2-dimensional. That is, it's really a curve, and maybe there are a little bit of like fluctuations around that curve. But as you go to higher and higher dimensional spaces, those fluctuations can go in different directions in the state space, meaning that your dimensionality keeps increasing. And then it becomes a more and more complex picture. But there is kind of a common mode that thinking about a 1-dimensional curve that has a little bit of noise in it and twist and turn in different locations, I think is an accurate picture, or as accurate a picture as I can tell you at this moment. The results are largely robust to the dimensionality reduction technique or n-dimensional state space analysis.
I think you have time for one quick question. We are close to the end of the session. Does anyone have a question? Please ask. Online question. Okay, go for it.
I will recite the online questions to you. First of all, Behnam has asked, looks like the baseline activities in the case of motion and phase task are different, around 19 for phase and 23 for motion. Maximal activity is also different. It seems that something is controlling the gain that LIP receives from sensory areas. Do you have any thoughts about this? Doesn't this go against your idea of energy efficiency, that is to preserve the firing rate while keeping it informative?
Beautiful question. That's a great question. So a little bit of difference is expected because these are not exactly the same population of neurons. So the neurons that contribute to different averages could be different. So a little bit of difference is explained that way. The point, so that was about the baseline, but the point about the peak is really important. So even when we focus on exactly the same cells that are recorded in 2 or 3 tasks, there is a meaningful change in the maximum firing rate that they can generate. There is also a change in the status space. So it's like, imagine if you have curves in a status space, and then these curves are rotated versions of each other, the changes that you are seeing in the averages would be expected. In fact, it would not be very easy. I mean, it is possible, but it would not be very easy to just get a curve and rotate it, but rotate it in a way that the maximum firing rates or the maximum differences would come out more or less the same thing. So this question that Behnam is asking is fundamentally related to the earlier questions that were asked about why rotation exists. And I think once we figure that out, maybe we will have a better idea for answering Behnam's question too. For now, I would say ignore the baseline differences, but focus on the maximum difference in the activity of right-left. And the best answer that I can give you at this moment is that rotations, if you just create random rotations, you would expect a little bit of change. But in what way that relates to the efficiency of the code or the representation of the or the implementation of the rotations, that's an open question.
Thank you. And there is yet another question. I will recite it on behalf of Dr. Ibrahimpour here in the conference hall. Do you think that it was a good idea for neuroscientists to go for RDM in the beginning? Don't you think going after faces or objects would be a better choice?
That's a great question. Yes, it could have been, but go back like 25 years in time, when, say, Shadley and Newsom were starting to record from LIP for the first time, and the links between psychophysical models of decision-making and neural responses were made. Newsom lab was doing direction discrimination. The initial ideas was that, well, you know, MT and MST have direct projections to LIP, so the next step to go and look at for how decisions are implemented is probably the regions that MT and MST project to. and LIP is at the top of the list. So they put their electrode in LIP and they saw what they saw, and the rest of it is history. So nice stratification based on a stimulus strength. Maybe that is a representation of the decision variable, and maybe LIP is representing the decision variable, and then findings in the other regions. So there was kind of a thinking process that was behind it. It wasn't a random task choice. Now, 25 years later, we have learned a few things. For example, we know now that the direct projections from MT-MST to LIP are really not information bearing for the decision-making process. They are important for other things that LIP is involved in. For example, the focusing of the attention spots on specific locations, or providing like a very fast code that can guide orientation responses and attention responses. The path that the information would ultimately use to get to LIP is also an open question, but most likely it involves subcortical regions and frontal regions involved in routing of the information. And that's also why the latency of the responses that we observe in LIP is, for example, 200 to 250 milliseconds in all tasks. Another thing that also has changed is that, well, now that we are looking at different tasks, we see that the neural code has important variabilities, and it's really not task invariant. I wonder sometimes that if the very first task that Shadlin and Newsom used for studying the decision-making process was the phase task, and if they had observed the reverse ordering of the PSTHs, would the link to the psychological models of decision-making be made so readily and so strongly, and whether the literature would find the shape that it has at this moment? I suspect that it would be different, but at the same time, because I've worked with both Mike and Bill, I know that they are extremely smart. And in fact, I want to refer to a conversation that I had with Mike a long, long time ago when I was a graduate student in the lab, that Mike was saying that, well, all that you really need for the representation of the decision variable is not the ordering of the PSTHs, but rather the separation of the PSTHs. Now, as long as there is separation of PSTHs, well, you can do any decoding and encoding that you want to do. That was way before the time that population activity and population analyses became commonplace and norm in the lab. But Mike already had the insights, so very likely they would have figured it out. But we can't really go back in time and recreate the history. All that I can say is it was lucky, and it was good that it happened, because if it had not happened, maybe there could have been challenges, and the literature and our thinking would be less mature at this moment.
Thank you very much, Dr. Kiani and everyone. Please join me again to thank Dr. Kiani. And we are really glad that we had you here this afternoon with us. With that, we say goodbye to you and hope to see you soon in person.
Thank you so much. Hopefully next time in person. Take care, everyone, and thank you so much for the great questions. It was an honor, and I'm so happy that I got a chance to interact with you, albeit for just a couple of hours.
Thank you.
Take care. Bye.